we have 4 keynote speakers or core talk speakers. And please use the chat space if you have questions from the speakers. And speakers, please use the chat space if you want to kind of answer the questions and keep the discussions active in the chat space. And one of my assistants, Payman, is already kind of posting the relevant literature to the presentations in the chat space. So I hope that we can keep the chat space as active as possible. The next speaker, Dr. Marom Bikson. Marom.
Hello, everyone. Thank you, Hamed, for organizing another amazing workshop. I'm going to be talking in the context of dose response about how low an electric field we might think would be physiologically active. And again, I'm just going to be sharing, this is more, I think, also like an opening talk theme. I'm just going to be sharing some thoughts. So, and I will put these slides online soon. So there, this equation, I think, is the only one I'm going to be talking about, and it describes how a compartment of a neuron polarizes when it's exposed to an electric field. So on the left, that term VTM is how much the transmembrane potential will change when the neuron is stimulated. So if it's at -60, it goes to -61. You would call that a 1 millivolt hyperpolarization. So that's on the left side of the term. This could be a dendrite or an axon. On the right side, you see the term electric field. That is the stimulation that neuron is being exposed to. So that will typically be in units of volts per meter or millivolts per millimeter. So for example, TDCS would produce half a volt per meter, whereas ECT or TMS would produce 100 volts per meter. And then there is a number that relates the two, just a constant, right? So if that constant was, for example, 2, then a 1 volt per meter field would produce a 2 volt polarization, or a 1 millivolt per meter field would therefore produce a 1 millivolt polarization. Okay, yes. So typically that constant is on the scale of about 0.1 for most neuronal structures. And so basically what that's telling us is that when a neuron is exposed to an electric field, it will polarize in response to the electric field in a way that we think could be approximated by a constant. Next slide, or move forward, please. The key thing to emphasize is that this polarization is compartment specific. And so this equation is per compartment. So a piece of a dendrite versus another piece of a dendrite versus a soma versus an axon will have a different constant A. So we need, and so that's why we get this notion that all compartments are not polarized similarly. Now, this theory has been exhaustively considered for different kinds of cell shapes, like starting with just spheroids or circles, axons that are crossing across the field, long axons that extend for long periods of time that may bend or may terminate. This theory seems to hold based on theories and experiments for different types of compact neurons. There are certain situations where the theory does not hold, and in those cases it is the change in electric field rather than the electric field per se that would be important, and therefore I would qualify that this theory is really under something I call the quasi-uniform assumption, which means that we can consider a neuron or a compartment exposed to one electric field. Okay, next slide. And so if you accept this theory, then you would say, well, there is no minimum electric field, because no matter how small the EF quantity is, because A is a constant, we will produce some form of transmembrane polarization, right? So no matter how low we go, we will produce some change in the membrane potential in each and every compartment forward. And because any change in polarization could therefore affect neuronal function, we get into this notion that, well, maybe there is no limit, right? No matter how small an electric field I apply, a compartment will polarize a small amount, and I can never discard a small amount of polarization as being irrelevant forward. And a lot of people have considered how small changes in polarization might lead to big changes. Next slide. And so then this leads in animal work to something I'm just going to call the how low can you go game, right? Where now you have a bunch of animal studies where you have exquisite control over the electric field that you're applying. You can measure the outcomes. And one thing that you can do in many of these animal preparations is you can apply the electric field hundreds of times, thousands of times over and over again. So as a result of that, you have tremendous signal to noise. You have the ability to resolve very small effects, right? So I have some outcome measure I'm measuring, no field, field, no field, field, no field, field, right? You do this hundreds of times to try to extract whether there is a statistically significant effect of the field. Forward. And there's been several efforts on this, including from our own labs. You can show that 1 volt per meter is active. You can show that half a volt per meter is active. You can show that 0.2 volt per meter is active. Forward. And I also want to flag up that by the same logic, you can design experiments to show that things don't work, right? We've seen these papers that say, this field, these fields are inert. Well, just like you can design experiments to resolve very, very low intensity fields. If I wanted to design an experiment to show that 1 volt per meter does not work, I could design that experiment as well, right? By having a noisy signal and leaving the amount of repetitions. So all this is to say that, the how low you go game is interesting, but maybe it's not going to teach us what we think it's going to teach us because it has this sort of experimental game component to it. So next slide. So I think that when we think about, and we're going to be hearing several speakers talk exactly about this, about what animal studies can teach us about how low intensity fields specifically can explain the effects of things like TDCS and TACS. I don't think we just want to see, is there an effect that we can statistically resolve for the purpose of publication, right? I think instead, we want to ask, what do these studies explain about how stimulation works? So for example, whereas traditionally neuromodulation was thought to pace action potentials, which is correct, I think, for things like DBS and TMS and ECT, for low intensity, maybe that's not the case. We're going to be hearing, I think in the very next speaker, about oscillations, right? So we can ask why are oscillations, or how are oscillations especially sensitive to electric field? Forward. We can ask, what about neuronal plasticity? Are there situations where the brain is undergoing plasticity, and because of that, it becomes exquisitely sensitive to perturbation by electric fields, especially small electric fields? Next point. We can ask where specificity comes from. So often when we stimulate, right, we want to affect alcohol craving, right? But we don't want to affect, where did you park your car, right? And so how can we use animal studies to ask the question, where can we get specificity from? Concepts like functional targeting. We can use animal studies to ask the question of when increasing more intensity does not produce more outcomes. Forward. I think just a few more questions. I think there's also some questions about superlinear sensitivity. So these are situations where we're seeing that seem to exceed what we'd expect based on the theory above. So we're seeing situations where we're either getting much more polarization than we thought we would, or we're seeing outcomes much larger than we would think based on the theory of polarization. And animal studies, we're going to hear this from different speakers coming up, can explain that. And finally, I think we can ask questions about non-neuronal stimulation. So the theory above applies to non-neuronal elements as well. I talked about spheroids, so it applies to endothelial cells of the blood-brain barrier. It applies to astrocytes. They're all being stimulated as well, perhaps with a different dose response. And so now when we're seeing outcomes of stimulation, something that lasts 20 minutes, we can ask, is that 20 minutes effect due to neuronal stimulation per se, or can there be other cells driving it? And so I think this is certainly, as we're going to hear from the remaining speakers, what animal models teach us. And I think these are more interesting questions than, the traditional, how low can you go, that and others, I think, sometimes focus on too much. All right, thank you. That was what I had to say.
Thank you very much, Marom. Thank you very much.